So hello and welcome to Biochemistry. Today, as the title suggests, we'll be looking at the modifications of gramstaining. So you all know about gramstaining. I have also wrote it down for your ease of understanding. So let's first understand what is gramstaining. Then we'll see where the modifications are done and by whom the modifications were done. So you need to remember these modifications and what exactly at what point the modifications are made. That's very important. Now, in case of normal gram staining technique, there is a primary stain. We all know that is crystal violet. Now, the decolorizer used here is 95% ethanol or acetone. That is the normal process I am telling. Whatever we are doing in the lab with normal gram positive and gram negative bacteria, it is used to differentiate between gram positive and gram negative bacteria. Now the mordant used here is the iodine or Lugol's iodine, and the counterstain is saffron. So for some seconds you have to put the crystal violet, wash it off, then put iodine, which will make a complex, crystal violet iodine complex, then wash it off with decolorizer, and then add the counterstain. That is the normal process. Now here, due to some issues with some bacteria where you cannot apply the gram stain or the gram stain results are confusing, that time scientists thought that it needs some modification for some organisms. Okay, now let's see three modifications done by, first one is the Kopeloff and Beerman's modification. Now Kopeloff and Beerman's modification, here the primary stain used is methyl violet. So the primary stain used here is methyl violet, that is PS, a primary stain. The secondary stain or the counter stain used here is basic fuchsin. So you can see the difference here. The primary stain in normal gram stain was crystal violet, which has changed to methyl violet, and later on the counter stain, which was saffrony in case of gram stain, now has been assigned to basic fuchsin. So these two are modified version according to Kopeloff and Beerman's technique of gram staining. Remember, these are also gram staining, only the reagents used in this reaction or the staining methods are different. Okay. Now coming to Jensen's modification. Here the decolorizer is 100% ethanol or absolute ethanol. So here we have used the decolorizer 95% ethanol or acetone. But in case of Jensen's modification, means gram stain, through Jensen's explanation, the decolorizer used was 100% ethanol. Now another one, the counter stain used here is neutral red. So first one you have seen primary stain and secondary stain, both are different. Here the decolorizer is different and the counter stain is different than gram stain. So it was discovered by, or invented by, you can say, Jensen's modification or Jensen's technique. Now the last one is the Brown and Brenn modification. It is a little bit complex technique, mostly used for actinomycetes in tissue sample. In tissue sample. So these actinomycetes present in the tissue samples are fixed by formalin. First modifications, formalin fixed. Now the primary stain is same as crystal violet, but the secondary stain used here is carbofuchsin. That is secondary stain, SS. And the decolorizer used here is acetone is to ethanol in 1:1 ratio. The decolorizer used here is the acetone and ethanol in 1:1 ratio. So here on another compound is also used, that is picric acid. Picric acid is mixed with the ethanol during decolorization. So this is a complex technique. The Brown's and Brenn's modification is a little bit complex as it is applied to the tissue samples, and there are some processes, the total step I have not described here. If you want a total separate class on this modification, I will do it definitely. So please tell me your opinions if you need the class of Brown and Brenn modifications. Other than that, that's enough for the modifications where you need to remember these 3 points. So I hope I have given you some new information, some useful information. So if you have liked my classes, carry on with me. And thanks for watching.